Where we left off, we were going over the VCF format. So I think what we wanted to do was finish those and then do some like variant calling and like actual data analysis. So let me go ahead and let's see, let me pull up my slides. Okay. Let's see. Go to presenter view. Show as tab. And then where are we? There we go. Hopefully now Zoom people can see my screen. Let me make it full screen. Let me jump way to the end. Okay, so brief summary of what we talked about last time, just to bring everyone back up to speed. So we talked about, okay, if we're in the middle of an epidemic, or if we're studying humans or some other species that has a reference genome, instead of us outputting the entire genome sequence, well, with viruses, we still do that typically just because it's small enough. But like with humans, I don't want to have to output the entire 4 billion letter long genome sequence of this new sample I've collected, when in reality, the majority of the genome sequence of that new sample is going to be identical to the reference genome. So instead, I could just output the differences with respect to the reference genome. So this is a task called variant calling. Computationally, it's the exact same thing as consensus sequence calling. It's just instead of outputting the complete genome sequence, we're just outputting the differences with respect to the reference genome. So just to briefly summarize what we talked about last time, the VCF file format is the format that you're going to be using to represent these. So the top double pound lines are the comment lines. These are header comments that tell you like what format of the VCF file specification are you using? When was this file created? Other relevant metadata that's useful, like what is the reference genome sequence? All sorts of stuff. Then these are all optional, but they're good to have. And most tools that output VCF files will typically have them. And most tools that use VCF files will typically expect them. But the first required line is this header line, which is just telling you what the columns are. I don't know why this is a required line when like you're not allowed to change the order. So it's kind of redundant, but I guess maybe just that the file self-contained. The first column is what chromosome you're looking at. So if we're dealing with viruses where you have a single linear chromosome, it's just going to be the name of the viral reference genome. But if you're dealing with like human, where you have a bunch of chromosomes, it'll be which chromosome you're referring to. Second column is what position you're talking about. And by the way, each row is its own variant. So yeah, second column is what position on that chromosome. Third column, I mentioned there's databases like SNpedia where you can look up specific special variants that might have some certain meaning. So if there's a special ID associated with this variant, it would be in that column, or dot means doesn't exist. The fourth column is the reference genome, like what the reference genome has at that position. So if we're dealing with a single nucleotide variant, so like a substitution or something, the reference is typically going to be a single letter. Or if it's a single letter deletion, similarly, the reference genome will be a single letter. But you could have instances where you have multiple letters deleted. So the reference might be multiple letters. You could have like a duplication event. So still, you might have like multiple letters in the reference genome, and like the other versions of it are going to have like multiple copies or something. But yeah, whatever is relevant at that position, the reference genome. The next column is the alternate allele or alleles if your VCF file has multiple samples in it. So if you have multiple alternate alleles, they're going to be separated by commas. So for example, in this third row, this specific position of chromosome 20, the reference genome has an A, but in our data set, some of the samples have a G and some of the samples have a T. So we have multiple alternate alleles for that position, so we just comma separate them. And then in the case of a deletion, we used to have a letter in the reference genome, but our sample has dot. So the alternate allele is a dot to represent deletion. In the case of like, I guess one allele has a deletion of length 2 here, so GTC became G in one of the alternate alleles, or GTC became GTCT, so an insertion of one letter. But yeah, so weird stuff can happen. That's kind of how you interpret those. I mentioned last time that there's like quality scores associated with each of these variants. So in the same way that when we run a sequencing experiment, I mentioned the FASTQ file, you have quality scores associated with each letter, which says this is how confident the sequencer is that this letter actually is that letter. Same exact idea here, where based on the coverage, so like how many reads cover that position of your genome, based on the base qualities of the letters covering that position, and based on some other like probabilistic stuff that goes into this calculation, there could be a quality score associated with that variant call. So the quality of this set of variants on this row, I guess it's just one variant, but the quality of this variant is 29, whatever that calculation is. You don't have to worry about it, but whatever specific tool comes downstream would have to know what unit this is in, which it could be different depending on what tool you're using. But basically some metric saying how confident you are that this is indeed this variant. You could optionally have filters in your VCF file. So here we have two filters. One is a filter that is going to be called Q10. And it's a filter where we want to discard things that have a quality score less than 10. And we have another one, S50, that is a filter basically discard variants where less than 50% of the samples have this data point. So like have this variant or have data about this variant. So even if it's the reference position, maybe less than 50% of your samples even cover that position of the genome. In this case, none of them use that, but you could theoretically include that in your filters. And what the filter means, you don't like delete it from the file. It still exists in the file, but then any downstream analyses that you do with it know, oh, I should ignore any of the rows that don't say pass, basically. So it's just like a flag for any downstream tools to know, hey, ignore this row. I'm keeping it for the sake of posterity so I can look at it manually, but don't analyze it in whatever thing you're doing next. Okay, so I think this is where we had left off. So I'm going to pause here and ask, does anyone have any questions about what we talked about last time? Yeah, go for it.
Do you have to code for like the filters or like the quality or whatever, or can you just use something from like a library?
Good question. Yes, the question was, can you like code for these filters or would you be able to use something from a library? So there's a toolkit called VCF tools that I didn't include in your lab, but I think if I teach this class in the future, I'm going to add it to the lab because I think lab 4 is a little bit short. VCF tools, and there's another tool, BCF tools. They're two tool suites that you could do all sorts of manipulations on VCF files. So those would be basically, you could take an input VCF file that's spit out by a variant caller. Like I think on the lab you use lowfreq and freebase. You can take one of those VCF files and then run VCF tools and specify all these different filters you want, and then it'll output a filtered VCF file that has all the same information, but then includes these filters. So yeah, you typically won't do it manually unless like you go into a PhD program where you're going to be dedicated to VCF file stuff. And like maybe as part of your dissertation, you'll write a tool that will have to manipulate these. But most people just use an existing tool. And in general with bioinformatics, that's kind of how it is. Most people, unless you're like a researcher in the bioinformatics space, you're probably using off-the-shelf tools other people have developed. But down the line, maybe you'll create one of those yourself. Yeah, great question. Any other questions from folks about stuff from last time before I go to, yeah, go for it. Good question. Yeah, so the question was, for these alternate alleles, is there like some criterion or like some threshold or something for you to have multiple instead of just one? It's basically, and I'm going to get to these in a little bit, but basically these final columns are your actual samples. And it's just a matter of, does this variant appear in any of your samples or does it not appear? So every alternate allele that appears in any of your samples would show up in this column. So if I, for example, I guess in this case, both of these people have both G and T. I'll talk about how to interpret these columns in a little bit. But in theory, like in one experiment, if I include two samples and one has one allele, one has a different allele, and they're both alternate alleles, this column would have both of them with a comma. But then if I like removed one of those samples, so I only have one of them and I only have one alternate allele, I would only show one value here. So it's just how many of your samples have, like how many alternate alleles show up in your samples. Yeah, great question. Awesome. Any other questions? And I guess a slight modification is also in the case of these like ID'd SNPs, you could theoretically look at SNPD or something like that, and they might have like predetermined alternate alleles that it could theoretically just fill in. Even if none of your samples have it, it might show up if it pulls it from some database. Yeah. but then they wouldn't really be used. Awesome. Cool. Okay, so that's the filter column. The next column is the info column, and basically it has additional metadata about this specific chromosome and position that is described up here in the header. So basically, one metadata ID is this DP, which stands for depth. So this is the number of reads that covered that position. So here DP equals 14 means there was a total depth of 14 reads covering that position. In this case, we have 2 samples, so I don't know if this means 14 reads across both of them or if it's the average of the 2 or something. I don't really know how they calculated total depth from multiple samples, but and like output it as a single number. But regardless, some metric of like how many reads covered that position. And the second one is allele frequency. So it could be allele frequency in your samples if you only have information about your sample. So how many, like in how many of your samples did this alternate allele exist? Or if you have access to some database, it could be across the entire human population, what proportion of people have this alternate allele. So you'll notice for the ones that only have one alternate allele, you only have one allele frequency versus for the ones that have 2 alternate alleles, they have 2 allele frequencies. And in this case, given that these 2 add up to 100%, I'm assuming these 2 are probably, oh, there was a third sample too, but I cut it off. So it should probably be 100%, either way. But somehow it's a metric of like, what proportion of the population has this, what proportion of the population has this, or it could just be constrained to your samples. And in this case, I had a third sample that I cut off that now these numbers don't really make sense. But in theory, that's what it would have meant. Yeah, so how frequent is this alternate allele? Okay. And you'll notice some of them are missing this information. So it's kind of optional for any of these individual variants to include it. But if they do, you have in the header comments what they mean. Okay, questions, comments, concerns. Okay, so all of this stuff so far has just been describing this variant position in general. But then the next columns are going to talk about your actual samples. So this column over here is going to set the stage for all the subsequent columns, where the subsequent columns are the actual samples that you're going to have. So this column format is describing every column coming after me is going to be a sample, and here's how we're going to denote what the variants are in that sample. So you'll notice it's like a bunch of 2-letter symbols separated by colons. So basically each of our samples is going to be in this exact format, where it's going to first be this GT entry, followed by a colon, followed by the GQ entry, followed by a colon, followed by DP, colon, HQ. And what do those metadata values mean are described here. So GT is the genotype, GQ is the genotype quality, so some quality score of how confident we are that this sample has that specific genotype. DP is the read depth, so that's like for the single sample. So I guess maybe total depth is the sum across all your samples versus read depth maybe is the individual. And then HQ is haplotype quality. I'll talk about what haplotyping means in a second, but don't worry about what haplotype means, just that is yet another metadata value, haplotype quality. And again, you'll notice not everything has all of them. It's just that if this row is going to have certain metadata values, the format column is going to specify what is the format of this row. Okay. Everyone still with me? Oh yeah, go for it, go for it. Yeah, good question. The question was, what's the difference between the genotype quality that's for this individual sample versus this quality score that was over here? So, and this take with a grain of salt, because I'm not an expert in this space. My understanding is that this is some quality that has to do with how we called this entire row. So across all the samples, what is the quality of us being confident that something is going on in this position? I don't really know how you interpret that. I think probably has to do with like this is probably some function of these individual genotype qualities, but I'm not 100% sure. But basically, this is some overarching across all of our samples. What is the quality of this position on this chromosome versus these are how confident am I that this specific sample has this specific genotype? Yeah, great question. And I think it depends kind of on the context. So like downstream tools, you would have some specifications of like how they interpret these, and then they would kind of react accordingly. Yeah, great question. Awesome. Okay, so the subsequent columns represent the actual sample. So this would be one person, this would be a second person. So this VCF file has variance for two people. How to read this? Well, the first column was the genotype. Maybe I should have also colored the genotype row here, but this was the actual genotype. So 0 denotes reference, and then the non-zero numbers refer to the alternate alleles. So here, 0, 0 means on both chromosomes, this person has the reference allele, so GG, versus like 1, 2 means that this person has alternate allele 1 on one chromosome and alternate allele 2 on the second chromosome. So 0 means reference. Anything non-zero is like the alternate allele kind of in that order. 1 is the first alternate allele in the comma-separated column. 2 is the second one, and you could have multiple if you have multiple samples. Okay. So yeah, so 0 means it's the reference, 1 means it's the first alternate allele, 2 means it's the second alternate allele. So you'll notice here that in the header comments, we have this line that says phasing equals partial. So has anyone heard of phasing before? Has this term ever come up? Or like haplotyping? Or like what a haplotype is? You might have heard in like genetics. No. Yeah, go for it, go for it. I mean, guess is totally fine too. So you said haploid. So what is haploid? So diploid versus haploid. Okay, so yeah, so haploid is you have one copy of your chromosome versus diploid is you have two copies of the same chromosome. So it's a similar source. So haplotyping, so when I do a genotype, let's say that I say your genotype is GA. So at this chromosome, this position, one of your chromosomes has a G, one of them has an A. Now let's say at another position on the same chromosome, you have like AT. Which letters go with which letters? So genotyping just means one chromosome has one letter, one chromosome has the other letter. But across the variants on that chromosome, I don't know which variants are on the same exact chromosome. So when you talked about like in a genetics class, is BICD 100 a prereq for this or no? No, okay. So for folks that are going to take genetics later on, here's a sneak preview. So humans are diploid. We have two copies of a given chromosome. But imagine that like you have some gene where, and let me see if I can, haplotype is collection of genes inherited from one parent. Exactly, it's the collection of variants inherited from one parent. So imagine I have like, actually maybe I'll do it vertically just so that it's easier for me to read. So imagine these are my two chromosomes, and this is like some specific gene. And let's imagine that like at this, so let's say I have two different mutations that can break this gene. And let's imagine I'm trying to figure out, I know I'm heterozygous for that mutation at both of those positions. The two scenarios are, do I have the deleterious mutation? So let's say that like I have one copy of the deleterious mutation at this position on this chromosome, and this chromosome is totally fine at that position. And then at this other position, I have another deleterious mutation in that same exact gene, and the other chromosome is also totally fine. Versus, let's imagine a different scenario. Again, I'm diploid, so I have two copies of my chromosome, but what if the deleterious one was on one chromosome at this position, and what if at the other position, it was on the other chromosome? So my genotype for both of these positions is the same. I'm heterozygous, deleterious, healthy at each of these two positions. But are the patients going to be the same? No, how come? What's going to be wrong or right? What'll be different?
This is like an X chromosome, so
Let's assume that it's like 1 through 22, but imagine one scenario, both of these like gene breaking mutations are on the same chromosome, versus in the other scenario, the mutations are on different chromosomes. It would break worse in the second person or in the first person?
Second person has like a the second person has like a buffer of like having
Well, so let's imagine that this X completely, like having one of these deleterious mutations is going to completely nuke the gene. So even just one mutation of these two completely nukes the gene.
you can still read it.
You can still read the other side? Yeah, we're going to see. Let's say it's a dominant mutation because it's completely deleterious. It'll well, so dominant, wait, so dominant versus recessiveness has to do with like, if I have two copies of an allele, one dominant, one recessive, the dominant one shows, the recessive one doesn't. But let's think like even lower level than like dominant versus recessive. Just think this is a gene that's important for function. And if you have only this mutation, that gene is nuked, that copy of that gene. If you only have this mutation, that copy of that gene is also nuked. So if you even have one of these two mutations in that gene, the whole gene is completely nuked. There's a question or comment in the chat. It would depend on penetrance. Let's assume that like it'll completely nuke the gene if you have that mutation. Any other thoughts? Yeah, go for it.
Okay, so for the second person, the one with it on one mutation on like one side and one mutation on the other side, if either one of those nukes the entire thing, then that person is gonna be worse off because both sides are gonna just fucking gone.
Perfect, exactly. So actually, the second person, I'm going to repeat your answer just so that folks on Zoom can hear. So the second person is completely screwed. Basically, if this mutation, just having either of these two, completely nukes that copy of that gene, I have two copies of every gene in theory, right? So in this person, this copy of the gene is broken because of this mutation. And then the copy on the other chromosome is also broken because of this other mutation. So this person is completely screwed. Like they don't have any functional copies of this gene. Versus this person, both of these deleterious copies are on the same exact chromosome. So yeah, okay, it's like extra screwed up, but like one of those mutations was going to screw it up anyway. So this copy is broken. Whether it's because of one mutation or two mutations, this copy is completely broken. But they have a fully functioning other copy that's still there. So like if you think about like if anyone's familiar with sickle cell anemia, so sickle cell anemia is a Mendelian disease where there's like a specific position where if you're heterozygous versus homozygous, like if you're heterozygous or homozygous for the dominant allele, like you'll be healthy, but if you have both copies of it are broken, you're going to be sick. So you can kind of imagine like here, this is a situation where like this person has one broken copy, kind of think of like a kidney. Like this person has a really screwed up copy of this gene, but the other one's working fine. So they're actually going to be okay, or at least better off than this person who is completely screwed up on both copies of it. So first, does this scenario kind of make sense? Questions about the scenario? Okay, so how is this relevant to what we're talking about? So knowing how to distinguish between these two scenarios has clear clinical relevance. If I take a newborn baby and I sequence them and I figure out this is the case, then, you know, it looks like they're going to be fine overall. Versus if this is the case, this is pretty bad. So how can I come up with this level of information? This is called a haplotype. So a haplotype is information about which variants are on each copy of your chromosome. So here, like both of these genotypes are identical. These genotypes are broken healthy, broken healthy. But the haplotype would be broken and broken are on one chromosome, healthy and healthy are on the other chromosome, versus this one is broken and healthy on one chromosome, healthy and broken on the other chromosome. So that information about which variants are on the same exact chromosome is your haplotype. And the computational problem of determining haplotype is called phasing. So here, let me I'll keep those diagrams there. So here it's saying that this VCF has what's called partial phasing. So for some of the variants, it was able to determine that information, and for other variants, it was not able to. Specifically, the vertical bars in your genotype represent phased position. So you can kind of imagine they're like bars separating two chromosomes. So everything to the left of the bar is on one chromosome. So like, for example, this person has a G at this position, a T at this position, a G at this position, and a T at this position, all on one chromosome. And then on the other chromosome, this person has a G at this position, a T at this position, a T at this third position, and then a T at that fourth position on the other chromosome. And all four of those are together. Does that kind of make sense? So the vertical pipe means this position, we have a phased genotype. We have the haplotype. So we know which variants are on which chromosome. If it's unphased, so this is a partial phasing VCF file, so some of the variants are phased, some of them are unphased. If it's unphased, we represent that as a slash. So the slash means this is your genotype, but I don't know your haplotype. I don't know, is this 0, which is GTC, is this on this chromosome? Or is it on this chromosome? I have no idea. So typically the unphased genotypes, we just write the numbers in numerical order, just arbitrarily, because we don't know the order, so we just arbitrarily say, okay, well, you're 0, 1, you're 0, 2. Versus for these ones, the phased ones, it's specifically which variant is on which chromosome. So the order matters for these ones, but for the unphased ones, they're just arbitrarily in sorted numerical order. Okay, questions, concerns, comments? Kind of makes sense. And again, typically you're not going to be playing with these by hand. You're typically going to be feeding them into some downstream tool, but it is useful to be able to understand them so that if you're ever doing like error checking or if you're trying to interpret the results, you can kind of make sense of what this is showing. Awesome. And I don't think I have any other slides. I didn't highlight these other columns, but basically genotype quality, these are arbitrary genotype quality scores. Depth, this is how many reads covered each of these positions in these samples. And then HQ, haplotype quality, you'll notice only exists for the column, for the rows that are haplotyped. So the ones that have the vertical pipe. This is some quality score saying how confident am I that this is actually the haplotype. So how confident am I that this chromosome actually has a 0 versus this one? I guess in this case, it's not interesting, but here, 1 and 2, how confident am I that that's actually the haplotype? Okay. Then I think, yeah, there we go. Okay. And you'll notice there's two numbers because it's like, how confident am I of like this one on this chromosome, and then how confident am I of this one on this chromosome? Okay. Questions, concerns? No? Okay, so I think some folks asked if I could do some like actual data analysis. So I was gonna probably spend the remaining 20 minutes like taking a raw sequencing data set, mapping it to the genome, and then doing variant calling. Is there anything else folks want me to do instead or in addition to or anything like that? Yeah, go for it. Ooh, gene set enrichment analysis. I don't think I can go over that today because it's not related to this week's topic. But when I get to RNA-seq, remind me and I can talk about it. Yeah, that's gonna be like week 9, sorry. But you can post on Piazza. And then if you come to lab hours, I think probably it's gonna be relatively empty just because I think a lot of people finished lab 4 already. But you should all go because you should get help on the midterm problems. But you can also ask the TAs. Awesome. Okay, so let me go ahead and let me stop sharing. Let me get all my stuff organized. So what I'm gonna do is human data is huge. So I'm gonna probably just take a viral sequence data set. Let me pull up. Maybe it's hard for me to like do second screen for this. I'm gonna just do a single. Oops, that's now only Zoom people can see. Let me duplicate mode. There we go. And then let me show my whole screen. Let me pull up Prairie Learn. So I'm going to do it directly in Prairie Learn just because you all already have these tools accessible to you. So if you go to the Unix playground on Prairie Learn, you can kind of, yeah, just play around with it. In week 10, I'm going to be dedicating the time to showing you how to compile these tools on your own computer. So in the future, if you're interested, feel free to let me know. Also, if you want, feel free to come to lab section and they can help you set these tools up if you want to. Brave browser, yeah, I just switched over. It's a good one. I'm digging it. My Chrome was crashing, so I was like, okay, fine. I'm gonna finally cut ties with Google. Ooh, dark mode. How fancy. Okay, let's see. So I'm gonna go ahead and open up a terminal. There we go. Awesome. Okay, so let's make this big. And then let me, what's the best way to find a data set? So let me go to, let's Google SRA. So SRA is the short read archive, or is it sequence read archive? Sequence read archive. So I'm going to search for SARS-CoV-2 just to see. And then here's one that I'd already found. Maybe I'll paste a link to it in the chat, and then folks can just follow along. So I Googled SRA, and then that took me to the short read archive. Then I just searched for SARS-CoV-2, and I'm going to arbitrarily pick this first one. So I'm going to paste the link in the chat for folks on Zoom, and then hopefully folks that are in person can see it or follow along. The accession number is like SRX something, 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 something. So I'm going to, whoops, yeah, so that's the result. So the sequence read archive has a bunch of raw data from a bunch of different sequencing experiments. And this run section is the actual sequencing run. So sometimes you'll have an SRA entry that has multiple sequencing experiments. In this case, it seems like it only had a single sequencing experiment. But regardless, I don't really care what I'm picking, so I'm just going to pick this one arbitrarily. So I'm going to click on this sequencing run. And it's going to take me to this metadata page about this sequencing run. You'll notice that it has information like what is the ID of it. I don't know what spots means. This is the total number of bases sequenced. So if I add up the length of all the reads, that's what I would get. This is the file size, GC content. These are things that you calculated back in week 1. When it was published, I guess they have a quality graph. I don't know what that means. Oh, so these are the base qualities. And then some other metadata about this, blah, blah, blah. But what we're going to do is we want the raw data. So we want the FASTQ file. So I'm going to click on FASTA/FASTQ download. And then taking a look. Okay, so I'm going to just download the FASTQ file. Starts downloading. Actually, I don't know how big Prairie Learn lets me save files, because I think this is like 100 megs or 30 megs. Oh no, this was like 30 megs, so that's not too bad. It's downloading. How fun. Why is it going so slow? This is like kilobytes per second. Oh, geez. Oh, really? Uh-oh. 600. Oh, kilobytes per second. Okay, I thought you meant like in this whole time it downloaded 600 kilobytes. Oh. Oh man, I mean, you all probably grew up in the era of like megabyte per second internet, right? Even when you were a kid. So I grew up, I remember our first internet connection was dial-up, and it was like 50 kilobytes per second. And then I remember we like upgraded to like cable internet, and it was 200 kilobytes per second. And that was like being hip. I could like download so much with 200 kilobytes per second. I remember I would like, I guess it's being recorded, but I would download movies legally And you would like set it running and it would take like days to run. Oh, good times. Okay, let's see. Oh, geez. I didn't want to open another terminal. So I downloaded that file. Downloading from SRI is always kilobytes per second. Ooh, that's rough. I wonder why. So I'm going to just upload that file that I just downloaded. Oh, sorry. Oh, so in the file browser, I right-clicked, and then there's an upload button.
You don't have to unzip it?
No, we don't have to unzip it. Yeah, so I would actually recommend probably not unzipping it so that it stays compressed. And then most tools that support FASTQ files will support gzipped FASTQ files as well. Like usually just by default, people keep them gzipped. Downloading movies legally, exactly. Law-abiding citizens here. So yeah, so I had just right-clicked in the file browser and I clicked upload and then I picked the file and then I uploaded it. So now it's in my environment and then in my terminal, let me get this up. I can see it here. If I want, I can use zcat to take a look at it. So I'm going to zcat, which is going to just concatenate it to standard output. Zcat is the same as cat, but for gzip files. And I'm going to pipe it to less just so I can kind of like take a look. It looks like it's wrapping around because this sequence is taking more than one line. So I'm going to pipe it to less space dash capital S. So the dash capital S means don't wrap around. Let it go off the screen. So now you'll notice each line has like a right arrow next to it, which means that I can like scroll right and left to see the rest of it. So now, as we expected, each entry is 4 lines. It looks like what we talked about in the past, where ID line, sequence of the read, delimiter, and then quality scores. It looks like these are already trimmed because these ones are 1 length. This one's a little bit shorter. These ones are much shorter. So I think these reads are already trimmed. So we mentioned back in, I forgot, was it week 1 or week 2? But we talked about trimming. So normally you trim the low quality bases from your reads. Today, we're not going to do that because it looks like these are pre-trimmed. But in theory, if they weren't like trimmed already, we would use FastP or something. Okay, so what's the first thing that we want to do? So first thing that we want to do is map against the reference genome. So here, this is a SARS-CoV-2 data set. So if you Google RefSeq is the NCBI database that has reference sequences. So if I search RefSeq, and then I can type in SARS-CoV-2, and that's going to search for the reference genome for SARS-CoV-2. Let's see, reference genome, severe acute respiratory, blah, blah, blah. Yep, that's what I want. And I've been dealing with SARS-CoV-2 sequences for way too long, so I know off the top of my head, this is indeed the correct reference sequence identifier. So let's download a FASTA file. How do I download it? Oh, send to, that's what it was. So let me go back. So to download this, it's not the FASTA button. That's just going to view a FASTA. You can do send to, complete record, file, and then for format, you can specify FASTA. And then create file. It's going to download the FASTA file. For my purposes, I'm just going to, oh geez. How do I get not big again? Small again. Darn it. What? Get small. Whatever. Okay. I'll just download it. Is it always going to be big now? I hope not. Oh, geez, I'm going to have to fix that.
Double click on the band all the way at the top.
Oh, I thought that was part of the other window. Okay. Whew, good catch. Thank you. Awesome. Okay. Am I becoming a boomer? I swear I'm a millennial. Okay, so there we go. We have this one. I'm just going to rename this to reference.fasta so I'm like familiar with what it is, but we can kind of take a peek and like, look, this is my SARS-CoV-2 reference genome. Okay, so when we have raw reads and we have a reference genome, what's the first thing that we do? Assuming that it's already trimmed, because we've already checked that these are trimmed reads. What's the first thing that we do? This was last week's topic. Any thoughts? So what file formats were the input files to your lab this week? They were BAM files. So what's a BAM file? It's alignment files. Yep, exactly. So the first step in any of these types of genomic experiments, if we're not doing de novo genome assembly or something that's directly working on the reads, which usually we're not, first step is map to the reference genome. So let's just use Minimap2 because I like Minimap2. So Minimap2 in general, you should always take a look at the help message. But the general usage is Minimap2. Target.fa is the reference genome. So that's reference.fasta. And then the subsequent entries are the FASTQ files that you want to map to it. So in our case, we only have one FASTQ file. It also supports FASTA files if you wanted to. So you can map like FASTA files against a FASTA file. But for us, we're doing a FASTQ file. So the next parameter is the FASTQ file. For our purposes, well, what do we want to do? We actually have to configure the input and output. So hopefully you remember way back in week 3, I guess that was just last week. From lab 3, you should hopefully remember what arguments you used, but we want to output in the SAM format. So we have to specify -A. And remember that read mapping is a heuristic. So we talked about the pairwise alignment algorithm that's super, super, super slow. And I ended last week's lectures by saying Minimap2 and all these read mappers do various heuristics to avoid doing that entire matrix filling. So the parameters that they use are going to change the accuracy of our read mappings. So Minimap2 has a bunch of convenient presets that you specify with -X. We're going to use the short read preset, so SR, because we have short reads that we're mapping against a reference genome. They have long read preset. They have like spliced alignment if you're doing RNA-seq for humans. You don't have to know what these other things mean. But yeah, basically a lot of different settings that they've kind of pre-optimized for different technologies. But we're going to use -XSR. Am I done? Should I run this? Should I do it? No one's going to save me? It's going to output it to standard output. So I'm going to get, let's see, it does some pre-processing. It's indexing the reference genome. What the heck? Come on. Oh, there we go. No, it's all going to standard output. So that's annoying. So what I'm going to do is I'm actually going to pipe to SAM tools. Oh, let's see in the chat, was someone going to save me, but you were just too late? Use arrow. So I could, so someone said I could use the right arrow operator. So in Bash, the right arrow means output to a file. And then if this is a SAM output, I could output to a SAM file. So like mapped.sam or something like that. I would actually, for Minimap2, suggest don't usually redirect to a file because they have a -O parameter. So if I run this with -H, is it going to show me the help? Yeah, so they have a -O. So if you wanted to like redirect it to a file, I would personally recommend just use the -O parameter. So like -O map.sam would have been the same thing. In our case, though, what we want to do, I think someone asked to do variant calling. So most variant callers are going to need your reads to be sorted. So you do the read mapping, the reads in our sequencing experiment, the sequencer closed its eyes and just randomly grabbed reads from all over the genome. But for doing variant calling, typically these tools need them to be position sorted. So after we've done the read mapping, we know exactly where each read hit. It's going to be some arbitrary order. And we typically want to sort them so that they're sorted by position. Yeah, go for it. Oh, is that why we have to index? So the indexing is so it's not why we have to index, it's we have to sort them because we want to index them. So the indexing is basically once they're sorted, we can run samtools index, which is going to create an index file that's going to specify like at this offset of the file, here is where this position of the reference genome starts. At this offset, here's where this reference position starts, so on, so that any downstream tools can like quickly in constant time, I guess not constant because technically it has to seek the file, but very quickly can jump to specific parts of the binary file. So yeah, we sort it so that we could index it to make downstream things much faster. So what I'm going to do is, since I know that I'm going to sort them anyways, I can actually just pipe this into samtools sort. And with samtools sort, I can specify an output file and I can specify a BAM file directly, and samtools will handle parsing the input as a SAM, and it'll handle writing the output as a BAM if I specify BAM. So for me, I'm going to specify a BAM file. So I'm going to say like mapped.bam. Minimap2 prints its status to standard error, which is why I still see it even though I'm piping. So remember, piping is going to take everything from standard output from one program and feed it to standard input of the next program. So in theory, I shouldn't see anything, but if I'm still seeing it, it's because these are getting printed to standard error, which is a separate output stream. But the actual SAM output is actually getting fed into SAM tool sort. So you'll notice here it's saying how many reads it's mapped. And I'm actually going to open a new terminal while that's going. I can open htop and I can actually see that, let me sort these. Oh, these tools are too fast. It finished before I could show you. But basically what I was going to show was the highest CPU was going to be alternating between Minimap2 and SAM tool. Actually, let me just do it again because that was so fast that it's a good learning outcome. Let me just remove it. Whoops. Run it again. So you'll notice Minimap2 is at the top because it's mapping, or I think probably right now it's indexing. So Minimap2 is using most of my CPU. And now you'll see SAMTool sort is showing up. And SAMTool sort is doing a little bit of CPU, but I guess it is less CPU intensive. Where did it go? Come back. Oh, there we go. It came back. So now it's like taking chunks of it, sorting. But I guess Minimap2 is the main bottleneck here. Oh, and now SAMTool sort is now the bottleneck, and then it ends. So by piping, they're kind of running simultaneously, where it's just feeding output of one tool to input of the next. Let's close out of that. Okay, so now we have our mapped BAM file. I could use SAMTools view if I wanted to take a look at it. So now, yay, we have our BAM file, which I can look at nice and cleanly. And you'll notice because I sorted them, so SAM format, first column is the name of the read. I think second column is a bunch of flags of some sort. Third column is what chromosome. And I think fourth column is it position? Let me see. Maybe, probably not. That's too many. Oh, these columns are probably position. Let's see. Wait, which columns are position? I always forget. Let me just jump to the end. Oh God, this is taking forever. Oh no. Oh no. That was oh, there we go. Oh geez, these are unmapped. Okay, there we go. I forget. That's okay. By the way, you see the SAM file. So, oh, we're pretty much at time. I'm just going to go ahead and finish this. So we can run viral consensus, which is the tool that I wrote, which this will do variant calling and consensus sequencing calling at the same time. It doesn't output as a VCF file, though. I should probably add that feature. But I can specify -I, take those mapped reads, -R, specify the reference genome, -O, and I can specify the consensus. So consensus.fastday. And then the position counts, this is equivalent essentially to variant calling-ish, essentially. So this would be like, let's say like position counts.tsv, and then -oi is insertion counts.json. I'm just kind of like copying this usage. Viral consensus runs. And it's done. So now I have these files. I can open up the consensus sequence, and we have our genome assembly. This is a consensus sequence. We have the position counts. So at each position, what are the counts of the different bases at that position? And we have insertion counts as a JSON file. But the main output here is the consensus genome. Yeah, go for it. How would you convert this to a VCF file? I have to write a script that I haven't gotten around to doing. But in theory, you should be able to take the position counts and the insertion counts and then output those as a VCF file. But I haven't done that yet. But if we want a VCF file, let's do that real quick in the last minute of class, or I guess we're already over time, but that's okay. Let's run lowfreq. How do I get the help? Oh. Okay, I guess if I don't specify any parameters. So one annoying thing about bioinformatics tools is everyone has a different way of specifying like how to get the help message. So I guess low freak is just run it without any commands. So low freak call, and what does that need? So low freak call, what's the simplest thing? I can specify a reference genome, so -f, whoops, reference. The output, let's say this is variance.vcf. And then regions, I want everything. Bed file we don't have. You don't have to worry about what that means. These things, I'll just use their defaults. Oh, and then the last parameter is the input file. So let's say mapped.bam. Low freak is running. So yeah, so if you wanted a VCF file specifically, just use low freak or something like that. But hopefully this summer I'll have time and I'll write a script that converts my output into a VCF. Someone asked in the chat, I was wondering why the documentation was so much easier to read when using viral consensus until I had written, yeah. So I'm a stickler on like writing clean, documented, and like help message code. Not everyone is, which is annoying, but alas. So all of you remember, you are your #1 user. So write well-documented code, because 1 month from now, you're not going to remember what anything is doing. So low freak is running. This is taking a while. You all can clean up if you want. I'll just wait till it finishes running. But I guess while it's running, any questions? Yeah, go for it.
Back to that BAM, and now it's got BAM file real quick.
Yeah, mapped.bam. Let me open up a new terminal. So you want me to look at it?
Yeah.
So samtools view mapped.bam, and let me pipe it to less-s. Okay.
I just noticed somewhere there was like a lot of nucleotides that were N, labeled N. What does that mean?
Yeah, good question. The question was, what do N nucleotides mean? And actually, the consensus output that we got also had that. So you'll notice these N's at the beginning. So in a FASTA or a FASTQ file, N is the ambiguous character that means this could be any nucleotide. So it means there was a nucleotide at this position, but I just don't know what it is. There's also other ambiguous characters. So if you search for IUPAC, maybe ambiguous. So there's these IUPAC ambiguity codes that are basically, so A, C, G, T are unambiguous. Those are those 4 letters. Y stands for a pyrimidine, so either C or T. R stands for a purine, so either A or G. W stands for weak amino properties or whatever, strong property. I don't really know what those mean. That's a biochemistry question. But W and S are A or T versus G or C. So basically every combination of which things I think it could possibly be, there's an ambiguous letter for that. And then N just means A, C, G, or T. Yeah. So let's see, is this thing done yet? Let's exit out of that terminal. No, this is still going. So I'm gonna stick around until it finishes, but if you want, feel free to head out. But basically output is gonna be a VCF file. But yeah, my tools are very fast. That's nice. Awesome.
You didn't look at what this person has done at all.
What Low Freak did? No. So I just create. So the tool that I based mine off is a tool called Ivar. So if you look for like Ivar Anderson Lab, it's not a very like easily found name because Ivar is also just a human name. Like it's just a name of a person. But yeah, so if you find this GitHub repo, so this was kind of the gold standard that people were using for viral sequencing. We were intending to do consensus sequences because with viruses, usually we do consensus sequences because they're short anyways. But during the COVID pandemic, basically like Rob Knight and me and like a couple other people like banded together to like try to speed up the sequencing pipeline in San Diego County. And this tool was too slow. So based on this, I wrote my tool from scratch to like not do the slow things that this tool was doing. But I didn't look at like variant calling tools because I didn't really think about it, but I should add that functionality to be able to output a VCF file, but just haven't gotten around to it. Yeah. How many people have downloaded it? Probably not a lot, to be honest. Let's see. How can I get like GitHub statistics? GitHub repo statistics. Can I find that easily? GitHub stats. Oh. Well. I can try git stats. Oh, I don't want to install a program. Oh, okay. Yeah, let's see. So github.com/nmst/viral consensus. Yeah, are they like, oh, insights? Let's see. So that's not traffic. Oh, 4. Yay.
It's 122 views of people who looked at it.
So not a lot. Yeah. Oh yeah, that's true. Yeah, that's probably you all. It's like half of this class. Oh, yeah. 39 out of my 40 unique views. Yay. And we have 99 people in the class or something, so that's not even everyone. Yeah, yeah. Oh, with Brew, that's a good question. So you could definitely do it with Conda.
You can see like 2 people from like some academic website.
Oh yeah, this is from the paper. So yeah, so people saw the paper, so that's good. Let me see. So I can look up on Conda. So Conda is like another one of these like package installing tools. How do I, is it Bioconda? Let me look up Bioconda. Bioconda search. I think I called it that. There we go. Does this have statistics? Not a lot. It's the fastest that exists though. Oh, that's true. Okay. That makes me feel a little bit better. This graph. Oh, okay. Oh, that's not bad. Oh, wow. Hey. Oh, no, 2,000. 12,000. I think this is every package, and this is showing where I sit compared to. But I'm on the upward trend. So smash that like button. I'm on my way to stardom. Yeah, I mean the problem is with bioinformatics tools, it's basically whatever people know about is the one that everyone uses. So I'm not as famous as the other guy, so then no one uses it. Cry.
Yeah, I feel like nature.
Yeah. Oh, we have like a bunch of nature papers, but that's true. I'll like artificially pump up my numbers. Oh, there we go. That's finished. Okay. Whew. Oh, you all will become professionals, and then we'll boom.
We should start a bioinformatics podcast.
There we go. Oh, there is one. I have a buddy that has a podcast. I should get on his podcast. Okay, so this finished. So now we can open up the VCF file. And just like we mentioned, here's all the header lines, this line, and then here is our, wait, where's our genotypes? Oh, wait, what? Where's our genotypes? Yeah, what the heck?
Well, then you're gonna make your VCF file better.
Yeah, so my tool is superior. Where's the genotypes? Yeah, what the heck? Yeah, shoot. Yeah, maybe that was a non-standard option that I didn't pick. But like, why would you run a variant caller and not want an output a VCF file? And then let's try Freebase. Freebase -f reference. And then let's do mapped.bam and then freebase.vcf. Oh, that's useful. Is Freebase going to take a long time as well? htop. Yeah, it's still running. Okay, while that's running, I can see why. Oh, geez. Why did this not output the base calls? Because it outputted some of the rows, but huh. Yeah, I'm not too sure. Maybe some of the settings are too high for this data set, so maybe this is not a super good quality data set. I don't know. Oh yeah, message. Let's see. To the moon, yay! I did. Yeah, just keep mashing it, that's true.
Yeah.
Okay, so this time it ran, so let's see. Freebase.vcf. Do we get variants? Oh, this has so much more metadata. Whoa. And we get genotypes. Hey, there we go. Yay. I don't know why it has like two genotypes though. This is a haploid. Oh, that was probably one of the let's see. It probably assumes so variant calling usually we do for humans just because humans are the ones that have the long genomes. So let me just do freebase -H.
It was gonna be a diploid, and you have to be like, no, it's not gonna be a diploid.
That's probably it. Yeah. So here there's probably
Oh,
oh
yeah, yeah,
oh yeah, pleidy. Oh, where?
Yeah.
Let me just
yeah.
yeah. Oh yeah, there we go. Yeah, so I should have specified -P1. Yeah. Now you all should go to class. So go to the lab section. The TAs are probably crying because no one's there. So be there. If you have already finished the lab assignment, feel free to get help on practice problems for the midterm. Feel free to like play around with this some more. Awesome. So yeah, let's go ahead and end the Zoom call. Thanks so much, folks. I will see you all on Monday. Bye, everybody.